हार्मोन इन्वॉल्व इन एनाबॉलिज्म। एनाबॉलिज्म में जो हार्मोन इन्वॉल्व होते हैं, फॉलोइंग आर द हार्मोन विच आर इन्वॉल्व इन एनाबॉलिज्म। मदरसी हार्मोन जो है वो एनाबॉलिज्म में इन्वॉल्व होते हैं। एस्ट्रोजन, इंसुलिन, ग्रोथ हार्मोन, टेस्टोस्टेरोन। Stages of anabolism. There are three basic stages of anabolism. Number one stage involves production of precursors such as amino acid, monosaccharide, isoprenoid, and nucleotides. And second step involves activation of these precursors into reactive form using energy from ATP. And third form involves the assembly of these precursors into complex molecules such as protein, polysaccharide, lipids, and nucleic acids. Sources of energy for anabolic process. Different species of organism depend on different source of energy. मुख्तलिफ species जो है वो मुख्तलिफ sources of energy पर इंहिसार करते हैं. Autotrophs such as plants can construct the complex organic molecule in cells such as polysaccharide and protein from simple molecules like carbon dioxide and water using sunlight as energy. ऑटोट्रॉफ जिनमें पौधे आते हैं वो अपना खुराक खुद बनाते हैं, कार्बन डाइऑक्साइड और सनलाइट को यूज़ करके वो अपने लिए अपना खुराक बनाते हैं. हेटेरोट्रॉफ on the other hand require the source of more complex substances such as monosaccharide and aminosaccharide to produce these complex molecules. Photoautotrophs and photoheterotrophs obtain energy from light, while chemoheterotrophs obtain energy from inorganic oxidation reactions. Anabolism of carbohydrates. In this step, simple organic acid can be converted into monosaccharides such as glucose and then used to assemble polysaccharides such as starch. Glucose is made from pyruvate, lactate, glycerol, glycerate 3-phosphate, and amino acid, and the process is called glucogenesis. Gluconeogenesis converts pyruvate to glucose 6-phosphate through a series of intermediates, many of which are shared with glycolysis. Usually, fatty acids derived as adipose tissues cannot be converted to glucose through glyconeogenesis, as these organisms cannot convert acetyl coenzyme into pyruvate. This is the reason why when there is long-term starvation, human and other animals need to produce ketone bodies from fatty acid to replace glucose in tissues such as the brain that cannot metabolize fatty acid. Plant and bacteria can convert fatty acid into glucose, and they utilize the glycosylate cycle, which bypasses the decarboxylation step in the citric acid cycle and allows the transformation of acetyl coenzyme to oxaloacetate. From this, glucose is formed. Glycan and polysaccharides are complexes of simple sugar. These additions are made possible by glucosyl transferate from a reactive sugar phosphate donor such as uridine diphosphate glucose to an acceptor hydroxyl group on the growing polysaccharide, the hydroxyl group on the ring of the substrate. Substrate can be acceptors, and thus polysaccharide produced can have straight or branched structures. These polysaccharides so formed may be transferred to lipids and proteins by enzyme called oligosaccharide transferase enzyme.